Good afternoon. I am Frederick Griffith, and I am going to be explaining my experiment in the year 1928. During the year 1928, pneumonia was infecting people worldwide, and I was trying to find a vaccine to cure it. In my research, I have found two forms of pneumonia that have been infecting people: the smooth strain, or S strain, and the rough strain, or R strain. The smooth strain has a smooth surface. The capsule around the cell wall protects it from various threats, and it is virulent. The rough strain is avirulent and doesn't have a capsule, which makes it susceptible to the immune cells of the host body. I began my experiment with 4 mice that I'm going to inject with different variations of each strain. The first thing I did was inject the first mouse with the S strain. As I thought, the mouse died, which means the strain causes disease and is pathogenic. I injected the second mouse with the R strain. This mouse continued to live, which showed me that the R strain was avirulent and did not cause the disease. The next test I did, I heated the S strain to be injected into the mouse. I then injected mouse 3 with the heated up S strain. After this part of the experiment, the mouse stayed alive. When I looked inside of the mouse, it showed that there was no bacteria in there. That means that the heat had caused the S strain to lose its capsule, which caused it to become avirulent. For my final test, I was going to inject the mouse with the heated S strain and the R strain at the same time. After doing this to the fourth mouse, it had died. This caused me to think about what had happened to cause the mouse to die. After taking a sample of the fourth mouse, which had died, I had found living S strain in the mouse. After doing this, I had concluded that something from the S strain had passed to the R strain that caused it to become virulent. I named it the transforming principle. I also predicted that this could be part of the genetic material. Hopefully this information will be useful to help someone find out why the transformation happened when I injected the heated S strain and the R strain into the mouse and caused it to die.